Hello, good morning, good afternoon, good evening, everyone. This is Josh Levitsky coming to you from American Journal of Transplantation Journal Club. And today we are going to be, we have a panel of experts to be discussing two papers that were recently presented in this TCMR series. The series is a TCMR program that is going through kidney, and now we're up to liver. And the title of this is TCMR and Liver Transplantation, Biopsy-Guided Approaches to Patient Management. And I'm Josh Levitsky. I'm a transplant hepatologist at Northwestern. And I'm joined by Dr. Alberto Sanchez-Fueo, who's a hepatologist at the King's College in the UK. We can go to the next slide. Our panelists today to be discussing two papers that were recently presented on this topic. The panelists are from areas around the areas of different interests. Dr. Dimitris is here with us today. He is a professor of liver and transplant pathology at the University of Pittsburgh Medical Center. Dr. Nancy Quan Man is a professor of surgery at the LKS Faculty of Medicine at the University of Hong Kong in China. And Dr. Jeff McCann is a professor of medicine at the Centenary Institute of Cancer Medicine and Cell Biology at the University of Sydney, Australia. So I'd like to welcome all three of you and Dr. Sanchez-Fueo to this panel discussion to talk about these two papers, and look forward to the discussion. I'm going to let Dr. Sanchez-Fueo briefly mention the two papers, and then we'll go into the different discussion points.
So hello, everybody. These are the two papers that were chosen for this journal club. And these two papers were uploaded into YouTube some time ago. So we are hoping that the audience had an opportunity to see the presentations by the PIs of the studies. So the first paper is the molecular diagnosis of rejection in liver transplant biopsies, the first results of the interliver study. And this was a multi-center study led by Phil Halloran in Edmonton, and the first attempt to implement this kind of a transcriptional gene expression tool, this molecular microscope that has been previously studied mostly in kidney and in heart transplantation into liver biopsies. And this is a peculiar study which follows the kind of a methodology of the previous studies conducted in kidney and heart, in that this had to do with kind of mostly for-cause biopsies within multiple clinical sites and without central pathologies. Now, the second study is a completely different type of study. This was a multi-center study from Hanover, led by Dr. Taubert, and it is one of the first attempts to use surveillance biopsies to guide the use of immunosuppression. Now, the reason why the two papers were chosen is because they raise what we think are very relevant topics in what regards the use of histology, surveillance biopsies, and how we manage mostly kind of at the development of subclinical liver allograft injury. And we've kind of put here in this slide a number of points that we think it would be worthwhile to discuss. This is in no means meant to kind of restrict the discussion, but just kind of provide some guidance of the topics that we think it could make sense to comment. Now, the audience can actually submit questions through the chat, so you can do that at any time. But I'm going to just stop sharing so we can see each other better. But just to start the discussion, I'm going to address a question to Jake Demetris and first, and then the rest of the panelists. And just for the sake of being a little bit controversial to start with, Now, this molecular microscope has been proposed mostly in kidney and heart as a complement or even as a potential replacement of conventional histopathology, under the assumption that the use of histology is not very reproducible and that a molecular tool could actually provide a more accurate diagnosis. Now, in the paper from the Phil Halloran group that is being discussed here, there are two references that are actually provided to support this claim that liver histopathology in transplantation is not very reproducible, but both come from the hepatitis C era. So the question, Jake, is do you actually think that the diagnosis of rejection, mostly in liver transplantation, is currently problematic now that we have eliminated hepatitis C? And how reproducible you think it is in highly kind of experienced centers such as your own, but also in kind of a less experienced centers? And then I would also like to kind of hear the views of the rest of the panelists.
I think it's highly reproducible, and it depends on your perspective and what your goals are, how you present the pathology. If your goal is to illustrate that another platform is superior, then you can find studies that say it's inferior. And I think that gets to the heart of the matter. The evidence that I can point to that suggests that it's reproducible is we just published a paper in Liver Transplantation looking at pediatric liver allograft recipients and protocol biopsies and for-cause biopsies done in that patient population. And the vast majority of histopathological parameters were in the high agreement category in terms of kappa values. And you can look at that paper, it's online now in Liver Transplantation. So, you know, I think molecular profiling can be helpful, but I think it has to be presented in context. And if you compare the data across the two studies, you could get some hints of where it comes up short. So we'll hear from other people.
Geoff and Nancy, the view from non-pathologists.
Yeah, I think I agree with the pathologist suggestions. It's always encouraging to see the add value for such molecular markers for actually on top of the as a traditional way to distinguish rejection pattern, right? Of course, I also agree with the feasibility and reproducible for such diagnosis ways, especially for the interliver study. Actually, this is only one-time biopsies for the not so large cohort, always multi-center. This with probably with some different confounding factors among the center studies. And they actually propose the different patterns. They claim actually these such diagnosis pattern can not only predict rejection, but also can look at the graft injury as well long-term graft fibrosis, right? I'm wondering actually, they only raise some the gene names patterns to indicate the rejections. So I'm not sure any study actually to correlate with the biopsy MR level linking to the circulated levels that would be more feasible to monitor the progress of rejection. Probably Josh has some ideas, right?
Yeah, I have to declare a self-interest because I was a co-author on the Halloran paper, and we submitted samples. I thought that the acute rejection signal out of this paper was consistent with what many pathologists have described, including Jake, and I didn't think that was controversial. I thought that there were the most, as Nancy suggested, I thought the most interesting aspect of the transcriptome was the separation of rejection from non-specific allograft injury in early, but also what I found and I'll be interested in Jake's response here, the fascinating result in long-term biopsies of the B cell signature and immunoglobulin signature in associated potentially with allograft fibrosis, and that emerged in later biopsies. And Jake and I have discussed with the Banff group many times about whether there's an aspect of antibody-mediated rejection in long-term liver allograft biopsies, and that wasn't addressed in the paper. We submitted some DSA data, but there was a paucity, total paucity of DSA data in the Halloran paper because most centers, excluding us, do regular DSAs. So I thought that was a fascinating aspect. The other aspect that that came out of the acute rejection signature that Phil Halloran mentions, I don't know whether it's mentioned in the paper, but certainly mentioned in discussion, that there was no antibody-mediated acute rejection signature, which was identified in kidney biopsies and heart biopsies. And I regret, Jake, that we had a couple of cases where we thought that there was acute antibody-mediated rejection, and we didn't get round to sending the specimens to Phil for the study. So I think that's unknown. I think to exclude acute antibody-mediated rejection in the first place in these biopsies would have required clinicians like myself to say, we think this is antibody-mediated rejection, and Phil's molecular analysis is saying, no, Jeff, you've got it wrong. And we didn't, and I think there weren't biopsies submitted to the inner liver study in that category. So I think the study confirmed the pathogenesis of acute T-cell mediated rejection for me, and raised interesting questions about the long-term aspects of graft function on a transcriptome basis. Yeah.
Yeah, I thought in Halloran's paper, it might have been more useful to completely divorce the comparison between the histopathology and the molecular signature. I had a very difficult time determining how they actually made the comparisons. You know, they used different cutoffs for RAI scores, starting with 1, 2, and 3. And, you know, most studies wouldn't include 1 and 2 in any kind of rejection categories. And even beyond that, there's, you know, comments about at many centers, there was no summary sign-out statement, and there was no statement whether rejection was present or not, you know, kind of in a binary classification. And with that uncertainty in the comparison, you know, no mention of biliary tract problems. They just, you know, the clinical comparison of the molecular signature with the biopsies just didn't ring true to my clinical experience. You know, no discussion of recurrent disease, no discussion of biliary tract complications, no discussion of vascular complications. So maybe they were lumped under the category of injury patterns, but I think it's probably more useful at this point to separate the two, or get a little more kind of granular or standardized comparison between the platform. And I also even had problems with the archetypal analysis. They said they based 4 sets, you know, and those 4 sets are, you know, if you're a lumper or a splitter, they generally ring true to me. But that was based on heart, kidney, and lung data. And then later on in the paper, they said, well, they found no molecular signature. Well, does that invalidate the archetypal analysis because they don't fit the phenotypes? You know, so there was a lot of comments that I really had, you know, trouble reasoning through. So I thought it might be better just to start from scratch with the molecular profiling and get rid of the pathology and say, these are the molecular phenotypes, and see how that works out. You know, that's contrary to what I usually do. I usually use all the information I have available to be in, whether it's translational research or whether it's patient management. I particularly don't use one tool. I use the biopsy findings, I use the DSA, I use the imaging studies, I use the patient history, and put everything together. So I prefer a cross-platform analysis. But if you're not able to do that in a rigorous fashion, I would suggest to separate them.
So Jacob, let me ask you just a little deeper question for that. So, I mean, I've sent you some cases, and your reads, I agree, are very detailed, elaborate, and go into all the features of a biopsy. I think a liver transplant pathologist may not go as deep as you do into the different aspects of the biopsy, and sort of read it as, just TCMR, present the RAI scores. And so the question, I guess, is, do you think in your mind, a molecular profile, let's say, again, the paper isn't perfect, but it does suggest there is a molecular profile for rejection versus late injury and fibrosis. Do you think that would be helpful in standard practice, and also sort of in some of those difficult cases where you've got some mixed pathology, particularly late after transplant, where you may have plasma cells, you may have some evidence maybe of ABMR pathology. Do you think that having a profile would add to what you're saying, your armamentarium of to help come up with a diagnosis for clinicians?
Yeah, I think targeted profiling can be very useful, but I think it only should be clinically implemented after more rigorous translational research, because the tests aren't cheap, and to just start doing this on every biopsy that comes through is going to be, a substantial waste of resources, in my opinion. And if you use a targeted approach, yes, well, we start to see these are problematic areas, and these are cases that would benefit from more information. But doing translational research and writing a manuscript is different, much different, as you know, than implementing a clinical test. Where I've seen the molecular profiling in other organs, I still think even in those organs, it needs more validation, because sometimes it can also mismanage or misguide patient management.
Of course,
Can I challenge Jake a little bit about the early biopsies? I mean, the early biopsies, I thought the transcriptional profile separated out acute rejection with TCMR profiles from other organs and interferon gamma approaches from other forms of injury. So I was less pessimistic about the validation of that, and the data that Jake asked for about exclusion of biliary injury and exclusion of arterial thrombosis, that was in the database that was all sent to Edmonton, but probably wasn't included in the paper. So I thought the acute, the differentiation of rejection, which I think Josh was correctly based on fairly superficial pathological analysis from all the centres. A little bit surprisingly, I think, but confidentially separated out acute rejection from early injury. I'd like to ask Jake how he interpreted the longer term B cell signature, because I think that can be interpreted two ways. One, it can be interpreted as, is this really an emerging antibody-mediated rejection signature in longer-term allografts with fibrosis, or is it associated with the fact that it's known that B cells are required for liver fibrosis in transfer experiments in experimental animals? So I'd be interested to what Nancy thought about that, and also Jake thought about the longer term signatures of B cells and immunoglobulin, and also endothelial signatures in later biopsies.
Either
Nancy,
one.
do you want to go first?
Well, I think it's understood that it's still far away to put this molecular diagnostic marker directly combined with clinical practice, right? But we do see the add value for this profiling, especially can distinguish different patterns. So the paper also, the author mentioned limitation. They cannot directly distinguish the T cell mediated response and antibody mediated response and rejection. But we do actually see different patterns, right? So as I mentioned, it's also worthwhile to look the follow-up, because the follow-up time after biopsy is not so long. So it is still worthwhile to look at long-term outcome and the different endpoint. So as I mentioned, when we look at the transcript gene levels, it's also worthwhile to look at the circulatory levels that match to the gene levels in the liver tissue. So that would enable us to monitor the circulatory level to look at the time course, actually the dynamic development for the rejection, as well as long-term outcomes such as the graft fibrosis. I agree with Jack. Actually, histological changes, especially different patterns, will be very important. I'm not sure the biopsy sample is enough to parallel to do in the core biopsy, in addition to the MRI isolation, and the plus the histological match, the detail, the imaging. So that's really interesting. And we also can look at the different histological, pathological patterns and compare with the gene levels.
Yeah, I thought the late profile was interesting, and I don't want to be perceived, and want to correct a possible impression that I'm anti mRNA expression profiles. We have a paper coming out on hepatology that we use them extensively. It's just that they should be implemented properly, and I think the best approach is a cross-platform approach. You know, I'm very strong advocate of using whatever data you can, because the goal is to solve problems in transplantation. The goal is not to say, well, this platform is superior to another platform. You know, pathology has its problems, molecular profiling has its problems, DSA has its problems, and you advance the field further and you solve problems by using a cross-platform analysis rather than saying this is the best, this is the most accurate, this never fails. When you use that posturing, you end up painting yourself in a corner, basically, that you're not going to get out of in the future. As far as the long-term damage, you know, if you look at the data that's been accumulated after the last over the last 3 decades in livers, things gradually deteriorate in general. That is a general truism. And I think that antibodies are involved. Do I have relatively good data? I think the data are starting to emerge that are pretty strong. All of the weaning studies that you look at, the DSA people, their grafts inflamed, they have more fibrosis, they don't do as well long-term, so there's smoldering injury. But one of the problems in the field with both pathology and molecular profiling, and it's not really their problems, it's a problem with the field and the grant funding cycle, is the delta that we're looking at occurs over a decade, and the grant funding cycles are 3 to 5 years. And by the time the grant funding cycle's up, I say, oh, you didn't find anything interesting. Well, it takes longer than that. And I think that's a deficit in the field in general.
Can I ask a question of, sorry, Alberto,
Go ahead.
yeah, so First of all, by the way, for the participants, there's a Q&A on the bottom of the Zoom. That's a place where you can ask questions. If you want to click that and pose a question to the panelists, please do so. But I wanted to ask Nancy. You mentioned something the first time you spoke about blood versus biopsy, signatures, and I think you said that the utility of peripheral blood signatures would be higher in patient management in terms of being able to do them serially, non-invasively. I sort of wanted to ask your opinion about that, but also how important is it that the signature in the blood is matching the signature in the biopsy? Because I know that in kidney and heart things where they've done this in other organs, they haven't really matched up very well in the tissue versus the gene expression in the peripheral blood. Do you think that's an important aspect to sort of, with these signatures, that there is a parallel gene expression in the blood or in the tissue, or do you think it's obvious that it's going to be different and that's just well understood and it's sort of we're catching things at different times? Obviously, biopsies are done infrequently compared to blood assays. So I just would like your comments on the blood biopsy comparison. I think that's what you mentioned initially.
Thank you, Josh. So I think the liver biopsy and liquid biopsy definitely is some discrepancy, right? But I noticed the paper actually some of the gene is linking to the inflammatory cytokine chemokines. So this would be worthwhile to further to explore the possibility to look at the tissue level as a blood level, right? But some actually the factors will be difficult to link to the circulatory, probably not circulatory proteins, right? So that's actually, you can see discrepancy between the tissue and the liquid biopsies. But if we can find something correlated well with the tissue, so it would be more feasible for clinical application. At least we can monitor at a different time point longitudinally to monitor the development of rejection of fibrosis or graft injury or even the cancer recurrence, right? So it's still worthwhile to look at the correlation both with tissue and the blood levels, I believe. Thank you.
Jeff or Jake, do you have any comments about that? Obviously we can see where the field is going towards commercialization of peripheral blood markers quite quickly, maybe too quickly. I'm just curious about and just coming back to Phil's paper, and Alberto has published also on tissue signatures, and Jake, you mentioned an upcoming one for your group. It's a busy field, so can we, the blood versus the biopsy, or is it really versus each other, or is it completely separate entity?
Yeah, I'm in the intrahepatic profiling camp, Josh, I'm afraid, and I think one of the interesting things that Jake's been measuring is looking at T cell antigen presenting cell interactions in the liver in rejection. So I think he's been able to identify cell-cell interactions which indicate an ongoing anti-allergenic response in the liver. But I can't remember from his data whether you see that in the absence of tissue injury. In fact, so acute rejection classically is defined by an anti-allograft response in the first place, but also a tissue injury response. So I'd be interested to hear Jake's comments about, is there a discrepancy about the detection of an anti-allograft response within the liver with very sophisticated high technology immunohistochemistry type approaches versus, a liver injury response at the same time? Has he been able to separate those two? I think the use of peripheral blood, it'd be wonderful to have peripheral blood signatures which were specific for rejection as opposed to other other aspects of allograft injury, but I'm not sure we're there yet. But I'm interested in this sort of, intrahepatic allograft response as opposed to actually injury.
This goes to the point of the evolution of techniques and the criticism that pathology is irreproducible, and the pathologists are often stereotyped, but they don't take into account the evolution of pathology. And there's very more sophisticated techniques that we can use. And what Jeff's referring to, it'll be coming out in hepatology. We looked at immune synapse formation in the lobule. And it's a concept that I refer to in that paper as pre-pathologic rejection. And when you give a presentation on it, people say, well, that's an oxymoron, pre-pathologic rejection. Well, it really isn't, because if you look at the slides by routine histology, they look completely normal and there's no damage. Liver function tests are normal. Everything is completely normal. But if you look at it in a more sophisticated fashion and quantitative using image analysis, you can pick up these immune synapses that can tell you if you, with a fairly high degree of accuracy, if you lower immunosuppression in these patients, they're likely to reject. And it is borne out. We have 3 studies now. 2 of them aren't published yet, but we did 3 different cohorts and showed that that occurs. And if I can be of helpful to any help to the molecular profiling field, this is anecdotal, but it bears out in the comparison of 2 studies. Look at the liver injury test profile in these 2 papers. In the Halloran paper, the normal ones, the ALT was in, I think, high 50s, 55, 57. And in the other paper, Talbert, they're bouncing around the 20s and 30s, depending on the pathology. So you're looking at 2 different groups here. You could say, well, maybe they're not comparable because, ALT and AST levels are different at different centers. But that rings true in many studies. All the weaning studies I've been involved with, the people that are really normal have very low ALT and ASTs in the teens and 20s. And even when you get up to the high range of normal. So how does that reflect? I went through with the nanostrength profile data with Alberto, all of the pre-weaning biopsies from the IWITH study. And as you said, Jeff, it's very good at picking up this blast of alloreactivity that occurs early after transplantation, the clear, mild to moderate that most pathologists would pick up. When you get into the smoldering rejection that occurs in all organs, kidney, heart, liver, where there's an infiltrate and there's mild damage, and the clinical data say you're not going to gain anything from treatment, but we know the graft deteriorates. In my experience, and again, it's an N of a couple hundred patients where I went through one by one, it wasn't so good at picking it up. So whether that's a sensitivity issue or whether you have to target your genes and lower the threshold or whatever to help in the situations where help is needed most. But again, the approach taken that, the molecular profiling is the perfect answer, and there's a certain cutoff, I doubt whether that's ever going to turn out to be true.
I think Phil Halloran only says that for grant applications, Jane.
Yeah.
So the second paper we've discussed, of course, takes a completely different approach. There is no molecular profiling in this study. It's just an attempt of using histology to directly guide the use of immunosuppression. So I wonder whether the panel could comment of whether they are convinced by this approach, whether they think that conventional histology can be granular and accurate enough to guide the use of immunosuppression using a surveillance biopsies schedule such as the one described?
I think it's already been proven that it is capable of doing that. If you look at the studies that we've done together and all the weaning studies that I've been involved with, you can use the histopathology, and prospectively it proves. If you look at the weaning study from the IWIF, just portal inflammation and interface activity predicted people that were going to reject after wean as a single parameter. And in that study, I don't want to be critical of the gene profile, but it wasn't able to categorize patients into clusters, and that's never referenced. The histology was, but then if you put the two together, and to make my point again, the cross-platform approach is the best. If you put these two papers together, you might have something very nice. If you say, okay, we did detailed histology analysis and we did detailed molecular profiling, and it makes my point that if you're a carpenter and you're building a house, they don't ask you want a saw or a hammer. You take all the tools that you need to build the house. And we're trying to solve problems in transplantation. We shouldn't be focused on one approach or limited to one approach.
Jake, but the papers you are alluding to, of course, these were papers of complete immunosuppression discontinuation and where the immunosuppression management was not directly linked to the histopathology findings. So what you are describing was a retrospective analysis of the outcome of the trial, which is not what is actually being used in Taubert's paper, where histology is being prospectively used, not just to stop, not to reduce immunosuppression, but to decide into how much immunosuppression to give.
Right. Well, I'll just remind you that the biopsies were all prospectively read. So it was a prospective study. I didn't know who was going to reject and who wasn't going to reject when the biopsies come in. When they were taken, they didn't wean them patients for a while. So that was all prospective data that was gathered. I didn't know the outcome. So it really wasn't retrospective from my perspective.
Geoff and Nancy?
I think the strength of the Taubert and Saunders paper is that this is in adults. Most of the data that Jake's talking about is in pediatric populations with a plan to withdraw immunosuppression. So this is in an adult population where they claim that the biopsy. I think the biggest signal that comes out of the paper is that people are traveling along, and I think in the paper, and I'm just looking at it now, that 70% of patients are on dual immunosuppression, and actually 25% of them are on triple immunosuppression. So, and these people are 88 months from transplant, so they're 1.5 years from transplant, and they're saying I think the major question they're trying to answer are these people over immunosuppressed, and I think the conclusion to the paper is, well, let's do a biopsy, and if the biopsy has virtually no inflammation and it has some inflammation, minimal BAMF maybe, but is essentially relatively normal, then you should be decreasing immunosuppression. I think that's the major signal. The question is that, I mean, whether biopsy helps you make that decision, because experienced hepatologists will say, well, if you're 18 months out and you've got an ALT of 25 and you're on triple or double immunosuppression, then maybe you should just, do you need a biopsy to decrease your immunosuppression? So I mean, I like the paper, and Jake's got me discussing this in Banff in a couple of months' time. So it's very timely for me to reflect upon it. So that's the big decision. But they did find, and I just can't remember the statistics. Maybe Nancy can help me about but there were a subsection of patients who were who did have significant inflammation where they did increase they thought increased immunosuppression was important. I haven't quite got my mind around that at the moment, though, but I'd be interested to what Jake and Nancy thought about it, and you guys thought about it.
Well, I agree with Jeff. Actually, the author also mentions this limitation because this is a single center, and this is only, well, the clinical practice, but not a controlled study, right? Jack also mentioned if we combine with the paper 1 and 2, that means the histology, pathology plus the molecular profiling would be perfect. Especially can aim the add value for especially with the profiling. For the prospectively, I'm wondering if the machine learning or artificial intelligence will help or not, especially when we look at particularly the molecular biomarkers, how can add value to the traditional histologically or pathologically diagnostic value.
So Jack, in the paper from Taubert, there is a proposal of a specific histological thresholds. Some of them are based, or the key ones are based on the table that was proposed in the 2012 Banff document, which of course was designed to identify patients who could be considered eligible for complete immunosuppression discontinuation. Which is kind of a different application of how this is actually being used in this particular paper. So do you think that we have enough evidence to be able to provide some sort of thresholds, either the ones that are proposed in the paper or alternative ones, again, not to propose to completely stop immunosuppression, but to propose to make adjustments to immunosuppression?
That's from my perspective and opinion, that's a hard question to answer because it depends on the patient population and the time of observations. And again, we go back to this grant cycle thing. And I thought the Weaning paper really nicely addressed it, and they acknowledged in the discussion that they don't know if this is safe because they have, 13, 18 months follow-up. And that's what you need to know if your threshold's safe. Those studies are very hard to do. The other point is the original disease. I give them credit. They included kind of all comers in that, PBCs, autoimmune, stuff like that. We all know from practical patient management, lowering immunosuppression in a patient with autoimmune hepatitis or PSC, you're more likely to get in trouble. So I give them credit. And that's why this is a hard question to answer. And when you get into weaning studies, it gets even more thorny because some of the investigators often see the pathologists on a power trip, that they're ruining people's studies because they're saying there's interface activity or there's problems with a biopsy, and it puts you in a very uncomfortable position. So if you're in a weaning study, the investigators want more lenient criteria. If you're in a patient management conference and you're looking at biopsies on a weekly basis and the biopsy looks the same way, they'll say, oh, this person's not very reliable on their immunosuppression anyway. He's just an unreliable patient. So it depends on the perspective and who's looking at the data.
I was this made me think of just sort of extrapolating this Taubert study and also Alberto's work on, how do we take these data and extrapolate it to our general liver transplant population? Because clearly most of the work that has looked at predictive biopsies and signatures have been in the withdrawal studies of very select patients. So, and obviously we are moving quite quickly towards non-invasive assessments like FibroScan. And I'm just curious, back to what Jeff said as well. We would, take somebody like this with a normal ALT and just reduce them. Based on these data and other data on FibroScans, do you think, where are we going with this? Are we going to start using, FibroScans or biopsies to manage patients in the future? I was impressed by Alberto's work on the absence of the normal stiffness, absence of TSA, and low ALT correlates very highly with the absence of inflammation on a biopsy. So do we need biopsies, or can we use non-invasive tools to lower immunosuppression, or do we just do what we've been doing for 20, 30 years? Where do you think this is going? That might be too big of a question, but I'm just posing that.
Yeah, I think it's a great question, Josh, and maybe Alberto can comment about the fibro. We tried to do a study and it got totally stuffed up by COVID of basically doing FibroScans all on our 20-year plus survivors with normal biochemistry, and we never got it done because of COVID. But do we have data on that? How many patients, particularly in either populations, because the long-term children, about what their FibroScan readings are if they've got normal liver biochemistry? I thought that's an important piece of information that I'm not too sure that we have, but Alberto may have it.
Yes, Josh is alluding to a paper we published last year in Journal of Hepatology, which was the analysis of the baseline protocol biopsies of one of our multi-center drug withdrawal trials. And in that study, we analyzed the baseline information, which include histology, gene expression, FibroScan, and donor-specific antibodies. And precisely because it was difficult for us to define a clear histological threshold of what it meant significant histological damage, we actually use as what we consider in that study our kind of a key endpoint, the high expression of the typical genes associated with rejection. So the same ones that Phil Halloran has defined in his interliver study. And using this as an endpoint, both donor-specific antibodies, transaminases, and FibroScan were actually quite informative in identifying the biopsies with a very, very high gene expression level. Now, this gene expression level, of course, correlated very nicely with the histological abnormalities that Jake has described, such as interface activity. But what was actually remarkable is that transaminases were an incredibly good biomarker, but only for really kind of what we would consider the extremes of what we consider normality. So subclinical inflammation was almost non-existent in patients with very, very, very low ALTs, and it was highly prevalent in patients with ALT in the range of 40. Where FibroScans and DSAs were actually most informative was in the in-between population, those with what we would consider average transaminases in the order of 20 to 30. In this population, combining either FibroScan or donor-specific antibodies actually was quite useful to stratify patients according to their likelihood of having significant histological damage.
I mean, the other important aspect of the study was the effect of kidney function and immunosuppression reduction. I mean, we know, we're dominated by the long-term follow-up of these patients now, and we know that sort of the long-term follow-up of these patients in terms of patient survival has not changed really significantly beyond the 1-year survival rate in 30 years. And so what was a little bit surprising in this study was that the reduction of calcineurin inhibitors at an average time of 18 months post-transplant led to an improvement in GFR. And you know, there's always been a bit of a concept that if you're going to change the GFR in these long-term patients, you've got to do it early. So this was changing calcineurin inhibitors a little bit later than that, but they got an improvement in renal function. I think that was a sort of secondary outcome of the paper. I think that should not be ignored. Really.
Yeah. The question that you raised, Josh, is the main thematic topic of the Banff liver meetings coming up in September. You know, what's the best way to follow patients? What do you need? And my perspective, it depends on where you're at and what your goals are. If you're at an academic center, I don't think the other techniques in isolation give enough resolution to really push the field forward. If you're being followed up routinely at a community center, then I think non-invasive approaches like the one that Alberto advocated, I think are perfectly fine. But I think saying to abandon biopsies, you're not going to learn anything. And I shouldn't say you're not going to learn anything, but for example, if you want to study the effect of DSA and the number of patients you need to enroll in studies is going to probably increase by log factors if you don't have a biopsy and if you're not looking actually what's going on in the graph. So the answer from my perspective is depends on where you're at, what your goals are, what kind of approach you need.
Well, Alberto, what do you think? We've had a great discussion. We weren't sure if this was going to go 20, 30 minutes or an hour, and we're almost at an hour. And there'd be a lot more to talk about, but I think we have to end here. But that was a great segue, Jake. The Banff meeting coming up in September. I think it'll be a lively discussion there on how to move the field forward. Do want to mention that we're going to be having round 2 of this, excuse me, TCMR program, basically discussing the same 2 papers. Alberto, can you put up the final slide? Is just for the audience in case they want to hear European perspective, and we're going to have Jake on again, but we'll also have Maria Ladano and Eleonora De Martin from Spain and France as co-moderators, so that we capture different perspectives and also areas of the world that can come in into these webinars at a better time for them, and that's why we have 2 of these. So the next one is July 26, 2020, coming up in 2 weeks, and this is, you can see the times here are more favorable for, I think, for the US and the European population, and the registration link is below. So we're excited to do this again. Thank you very much, Jeff, Nancy, and Jake, for a great discussion, and Alberto also for co-moderating.
Thanks, everyone. Enjoyed it.
Thank you.
Thank you.
Yep.
Thank
Thank you.
you. Thank you. Bye-bye.
See you.
Thank you.